Right, good evening, ladies and gentlemen, and welcome to this webinar offered to you by the Pretoria Medical Discussion Group, hosted by Ampath Laboratories. Before we start with the presentation this evening, I would like to give you some background around the event. The Pretoria Medical Discussion Group was started in 1960 by a group of general practitioners in Pretoria to ensure continuous medical education of healthcare professionals. Our speaker for tonight is an AMPAT pathologist and clinical virologist, Dr. Marieke Brauer, who obtained her qualification as clinical virologist at the NHLS, after which she joined the immunology department of Ampath Laboratories. And she has ever since played a pivotal role in the department and during the COVID-19 pandemic. Well, thank you, Dr. Marieke, and we can start with the presentation now.
Thank you very much, Janri. Okay. So in essence, I wanted to start off with the basics again. So the basics, we start off with the viral structure of the virus. And I'm pretty sure that most of you know this off by heart by now, but basically the spike glycoprotein is the main part of the virus that we will be discussing this evening. And this is the essential part that is required for entry of the virus into the host cell, and basically where the whole thing starts. And also, because this is such a crucial part of the viral replication, it obviously also would be a main target for the virus to change in order to overcome things like the host immune system. So we will be focusing a lot on that, but of course we also know that there's a nucleocapsid protein, and that's also forms part of some of our testing, and there's a variety of other structures. And just out of interest again, that the coronaviruses actually do contain the largest known RNA genome, which is also a very crucial factor when it comes to the actual mutation rates. So just to give you some background, and apologies, this is from the previous year's talk as well, but I just wanted to recap again what the issue is with coronaviruses and the whole case for rapid evolution of these viruses. So although we know that there is a high frequency of RNA recombination, that would typically happen if more than one virus actually infects the same host, they also do have an unusually large genome. And on top of that, it also has, or the family of coronaviruses also have a very large zoonotic reservoir and a host range. So we know that the whole origin of COVID-19 also originated with how easily it jumps from one host species to the other, and specifically in the case of the original SARS coronavirus from bats to other sylvatic animals. And it very easily adapts to new hosts. And we know that there's also documented transmission that happens from not only humans to domestic animals, but the other way around. So it's very adaptable. And we know that the chances of having novel zoonotic coronaviruses emerging is very, very good. And on top of that, of course, new variants of the SARS coronavirus too will very likely still emerge. So to date, we know that there's actually been a relatively small number of mutations, considering how many people globally have been infected by SARS-CoV-2. And this is related to the mutation rate, which we know occurs at a slower rate than some of the other RNA coronaviruses. So out of interest, it's about half the rate of influenza and a quarter the rate of HIV. And there's a very, very fine balance for these viruses when there is diversity that happens, and we call it a quasi-species, which would basically be a whole group within the species that have small little mutations that happen. And there's basically a risk of mutations occurring every single time that the virus replicates within the host cell. But it also needs to find a balance with replicative fitness. So in other words, not all the mutations necessarily are successful or beneficial to the virus. So it really needs to decide which ones to keep, which ones to let go. And interestingly enough, coronaviruses actually have an NSP14, which has proofreading function. And this is unique because most RNA viruses actually do not have proofreading function. But because it has such a huge and large genome, it really needs to proofread in order to make sure when it's going through its replication that it actually maintains its replication competence. And just some of the principles. So this would typically be what happens is that we have a whole group of viral population. So we really, when we are having an actual infection within a host, you probably have these small little mutations and very small variations in the viral genome in these different virions that are formed. But then you have a process of natural selection that actually also happens at the same time. So you basically have natural selection for growth advantages, and it would look for things like enhanced binding to the receptor, or faster replication, or a more effective suppression of avoiding the host immune response. So these factors all play a role when we're talking about variants. Just also, again, the basic principles. To point out that a mutation we refer to as an alteration in the genetic material of a cell or a living organism or a virus, and that it's more or less permanent, and that particular change can then actually be transmitted to its descendants. And we take as an example of how we refer to these mutations, that of HIV drug resistance mutations here, where the original letter basically represents the new amino acid, and the previous existing amino acid would be the last one, and the numbers in the middle is basically the position of that particular mutation within the genome. So you'll often see when they refer to the genetic the mutations of COVID, for example, that this is the same way that it's described. And we also know that there's been a lot of terminology that's been thrown at us in the past 2 years. And a lot of the times these terminologies are used interchangeably, but really there are minor differences. So where the mutation actually refers to the actual change within the sequence, a variant is then a virus where the whole genetic sequence actually differs. But it's a very imprecise term from a technical point of view. A strain, on the other hand, would be a variant where it actually has different characteristics that have been demonstrated, like, for example, transmissibility or antigenicity. And then we sometimes hear them talking about lineage. There, basically, we're looking at variants that have a few different mutations, but that all basically belong to the same lineage, which indicates the actual spread of a virus within a population. So, and we know that we've now gone through various different nomenclatures. When I did this talk last year as well, I was discussing the implications of having the next strain nomenclature versus the Pango nomenclature. The WHO have since standardized the naming, and they, on the 31st of March, have started to assign labels, which is according to the letters of the Greek alphabet. And I know a lot of people are joking and say that we need to move to something else, like perhaps the Mandarin alphabet, because we're running out of Greek alphabet at this stage. Then what is the working definition actually of a variant? So there are different terminologies, and the CDC also includes a few other variants. But the WHO goes with the variant of interest versus a variant of concern. So variant of interest would be when the predicted changes would be things like the viral characteristics such as transmissibility, disease severity, immune escape, and diagnostic implications or therapeutic implications, and that has been identified to cause significant community transmission with growing numbers. But a variant of concern would then basically be what meets the definition of a variant of interest, but also through a comparative assessment has been demonstrated to be associated with one or more of the changes, which would be an increased transmissibility or a change in the virulence or clinical presentation. So in other words, more severe disease, or if it actually decreases the effectiveness of the diagnostic testing, vaccines or therapeutics. And then, so this basically summarizes that. So there are certain concerning properties that we are looking for with a variant of concern to classify it as such. And that would be transmissibility, immunity, and also infection severity. And the comparator here would be your wild type virus, which is the original circulating strain or the Wuhan reference sequence. So let's have a quick look at what are the actual current variants of concern that we're dealing with. So these are the current ones. As you know, the first ones that emerged in December of 2020 was Alpha and the Beta strains or variants, and shortly after that, Gamma. Then along came the Delta variant, and that also coincided with South Africa's third wave. And then subsequently, at the end of last year, 24th of November, we heard about the Micron variant, and it was very quickly then classified to be a variant of concern, and since then really has caused havoc globally. Again, just to summarize, what are the main concerns with a variant of concern? So besides the enhanced transmission, the impact on severity, we also are concerned about whether that variant can actually be detected by diagnostic tests. And of course, whether there is actually a risk of reinfections, in other words, our natural immunity or our vaccine-induced immunity no longer is effective, and whether it, of course, then has an effect on therapeutic interventions. So let's just go back again to the structure. So again, to summarize that the spike structure really is the sort of main focus when it comes to the variants of concern at this point. And again, it's responsible for entry into the host cells. And we see that a great deal of these mutations affect our RBD, or the receptor binding domain. And that really is our main target also for neutralizing antibodies that are formed in the body. So it makes sense that the virus would benefit by actually mutating in those areas and changing those structures. Also then the spike mutations, we know finally that would actually lead to changes within the spike protein itself. And that would lead to modification of the actual epitopes for antibodies. And this could then impair spike binding by the anti-spike antibodies. An interesting thing, which is something that is not unique to SARS-CoV-2, but also to other infections that we've seen in the past, is that it's postulated that immunocompromised hosts may in fact contribute to the evolution of SARS-CoV-2. So if you think about it, that when someone is infected with the SARS coronavirus, in general, these infections are relatively short-lived, and the person would overcome the infection, hopefully in the majority of cases. So there is not that much time for the virus to actually evolve. But in an immunocompromised host, you often have this lack of CD4 T cells. And these actually contribute quite significantly to the neutralizing antibody response. So if there's a lack, you probably would have a lack of a proper neutralizing antibody response as well. And then subsequently, the person does not fully recover, and they actually do not clear the virus with a chronic infection ensuing. And then you subsequently get adaptation that occurs, and novel mutations can more easily actually emerge over time. Just to show you also, so this does not include the Micron variant. This is still some of the other variants of concern. But you can see, if we look at the actual genome of the virus, that the majority of those mutations do actually overlap or are in similar positions across the different variants of concern. And some of them are actually identical mutations that we find within different variants. And these all sort of occur spontaneously and just are of some benefit to the virus. and it actually enables them to then spread in the face of rising immunity within the population by maintaining their actual replicative fitness. So let's focus on Micron specifically. So we know that there are more than 30 changes to the spike protein alone. And this really is the variant of concern that really sparked a lot of issues, so to speak, because of this concern over so many changes within the spike protein. And there's quite a few well-known mutations also included in it, which we already knew beforehand would probably enhance its transmissibility. Also to mention that we now know that there are different sublineages within the Omicron variant. So the BA1, the BA2, as well as the BA3 sublineage. Now, BA1 was the original sublineage that we saw within South Africa. And I'll also talk about this a bit later on, but it's the one that we associated with spike gene dropout when we were doing diagnostic testing, and we would not be able to pick up the spike or see a very low level of spike being identified, whereas the rest of the gene targets in the PCR would come up positive. And this actually made it easier to identify that it is Omicron as opposed to, for example, Delta variant. The BA2, however, does not have that deletion, the same deletion than the BA1. And so this actually does get picked up by the S gene targets within the PCR assays. And so we don't actually see that fallout that happens with BA2. And interestingly, this variant or sublineage within the Micron variant is now on the increase, not only in Denmark, but also in South Africa. We have detected an increasing percentage of BA2 with our genomic surveillance. And then just to show you, this demonstrates quite nicely that the Micron variant really has a significant number of mutations. You can see at the top three bars there are the BA1, BA2, and BA3 sublineages. And they really are a huge number of mutations. And it also does include not only in the S1 and the RBD region, but also some around the furin cleavage site, which also has some advantages to the virus. So if we have a look at what happened from an epidemiological point of view and how quickly this virus really has displaced other variants, we can see here in South Africa that until about October, we saw a dominance by the Delta variant, which is the light blue. And then suddenly from November and December mostly, we saw a massive increase in the number of Omicron that was detected. And it was more than 95% by December. And then more recently, so this is in the week that was ending on the 22nd of January, we suddenly started seeing this red blip towards the end of the graph. And these are actually the BA2 lineages where we've seen an increase. And it was found to make up about 23% of sequences in January and is also still on the rise in terms of the percentage. And then what has also been noted very quickly when the Micron variant emerged is that there was a lot of talk about antibody evasion. Which is not surprising given the significant number of mutations that we see within the spike area. But basically, there's been a lot of investigation into this. And I'd like to draw your attention to this particular New England Journal of Medicine correspondence. So what was interesting here is this particular group actually had a publication earlier in 2021, and they made a polymutant spike protein, which essentially incorporated a substantial number of mutations that they incorporated into that. So it was basically designed that way for the purpose of a study. And they then looked at the neutralizing antibody responses, and they found that it was significantly reduced in that particular, it was called PMS20. When Micron then emerged, they realized that quite a number of the mutations actually correlate with the Micron variant and this PMS that was designed. And this gave them the opportunity to actually investigate and sort of do further testing in addition to their original study. And they basically showed that there was, they investigated what the response was in vaccinated persons. And they also found then that in those who had vaccinations and only had a 2-dose, definitely had a decrease in the efficacy. But then after a 3rd dose, they gained neutralizing activity against the Omicron variant. So there's a lot of new studies emerging around this topic, which then takes me to the next section. So let's just take a step back and have a look at what are the immune responses that we expect in response to COVID-19. So first off, we know that there is an innate immune system and an adaptive immune system. And the innate immunity, I'm not going to focus much on tonight, but obviously that also plays a significant role. However, it's not really associated with long-term protection. And we know that the adaptive immune system, on the other hand, which incorporates both your T cells and your B cells, which would be antibody production, and that has a more long-lasting immune protection or memory response. So let's focus first on the antibody responses to COVID-19. So we know that traditionally we would expect IgM responses, and especially in infections such as COVID-19, that's respiratory, an IgA response as well. And usually this would occur earlier than your IgG response. In COVID, we've seen that this response generally would be on an average of day 5 that you would be able to detect it, but it could only start later. But you also we've also noticed that there can be quite a short difference in the antibody development between IgM and IgG, as well as IgA and IgG. So it might be only a few days, or it may even be a concomitant onset. And a very paradoxical appearance sometimes as well, where we see IgG development actually prior to IgM development. And this is postulated to possibly have something to do with pre-existing immunity to prior coronaviruses that may play a role in that IgG response that we see. But it's not really certain. So there's variable kinetics with the antibody responses, and the appearance of the antibodies is very individually centered. So we've also seen that the IgM response, although it on average would disappear by day 50 or so, it can be detectable beyond 90 days after the infection. And also just to note that the severity of disease has also been shown to play a role in the level of antibody response. So in general, we find that there are lower antibody responses or even absent antibody responses in some cases, particularly with less severe illness. And with more severe clinical illness, we tend to see higher and more robust antibody responses. Also, just to note that we see very similar responses in terms of the immunity after COVID vaccination, but what really it should be aimed for, and something that has particularly been noticed with mRNA vaccines, is that you also see quite a strong T-cell response that kicks in following COVID vaccination. And we'll focus a little bit more on those responses as we go along. Also then, just to notice that the immune response that we see after vaccination, it's important also to note that not all immune or antibodies are necessarily neutralizing antibodies. And this was, of course, also the case after natural infection with COVID. The major target for the actual neutralizing antibodies, as I've mentioned before, is the RBD region of the spike protein. And ideally, we would have loved to do viral neutralization assays, of course, for everyone to see whether the antibodies that are formed are in fact neutralizing antibodies. But the problem is that these assays, viral neutralization tests, require biosafety level 3 laboratories because you're actually dealing with live virus. And they also are very time consuming and expensive to perform. So it's not really meant for high throughput and really is mostly used in research settings. So they have found that when they compared the antibody responses or antibodies binding antibodies to the RBD's region of the spike protein, that this tends to be the most reliable antigen to actually measure the amount of neutralizing antibodies, and that there is good correlation between neutralizing antibodies and the spike antibodies. Now, the durability of the immune response is another big question that is hanging over everyone's heads. So we know that the antibodies do form and that there is antibody response, but the question is, how long do these antibodies actually last? And I think a big question, of course, would be, especially after mild COVID, if the initial antibody response is mild, how long does that last? So we're going to focus a bit on not only the memory B cells, but on our memory T cell component as well. Now, firstly, antibody responses in several studies up to date have now been shown to be relatively stable for at least 5 to 6 months after infection with COVID. Now, having said that, this is still, or this is very much limited by our current experience with COVID and how long we have had COVID with us. There are some studies, and there's one particular study that I didn't have time to actually put into this presentation, but a recent publication by Ledro et al., and this was in the JAMA, and they found that the spike antibodies were detectable in persons after COVID infection up until about 20 months later. So what is not known in this particular study is what the correlation was for these persons with protection, and also no mention made of variants and the influence thereof. There is quite a few studies that have shown also that the spike IgG that is shown to be stable over about 5 months do correlate well with virus neutralization as well. And there's been some findings in another convalescent cohort in China after a 6-month follow-up where they looked at anti-spike as well as anti-nucleocapsid antibodies, and they've still found it to be detectable in 70% of patients. And then we also have to ask the question about the durability following vaccination. And this is quite complicated as well to investigate, because we are seeing more and more persons who had infection as well as vaccination then together. This particular study from Israel also caused a lot of concern in our communities, where they found that the initial COVID IgG levels after vaccination was actually much higher than what it was after natural infection. However, after 6 months, they found that 16% of the vaccinated subjects actually had IgG below the seropositivity, while only 10% of the convalescent patients had antibody levels. So there may well be differences in the antibody kinetics after vaccination as opposed to natural infection. So there's a lot of these studies that are still ongoing, and this, of course, prompts a lot of questions around the need for booster vaccinations. This study as well was quite interesting, which was done by the Johns Hopkins Institute, and they basically found when they compared antibody responses in persons who had COVID-19 initially and then was vaccinated, as opposed to persons who only had vaccination with no prior infection, that those persons who had natural infection as well had higher spike antibody measurements, and particularly in those who had 2 doses of mRNA vaccine then. Also interesting was 1 month after the second dose, they looked at persons who had COVID either 90 days before their first dose or closer to the actual first dose. And those who had COVID longer, more than 90 days before their actual vaccination, generally had higher antibody levels. And 3 months later, even higher, so 13% higher than the other group, as compared to the group that had COVID longer in the past. So there definitely is something that happens when there is a longer time period between actually getting COVID and actually getting the vaccination. And this all needs to be taken into consideration to sort of optimize the immune response when we give vaccinations in persons who had previous COVID. Not that I just want to mention, I don't want to advocate that you have to get COVID infection and to actually get a better immune response and then vaccination. Of course, we know that there's a lot more risks involved with actually getting COVID as opposed to getting the vaccination. But it does give us some idea about the breadth of the immune response. Then in terms of the vaccine responses and Omicron, so there's been some recent studies that have come out looking at specifically at Omicron and what the actual protective immunity is from previous vaccination and whether it actually still is effective. So we know by now that there's been studies that showed that 2 doses of the mRNA vaccine would actually decrease to less than 40% within a few months after persons have had the second dose. Whereas if those persons get a booster dose, they get a 60 to 70% protection from the infection from about 2 weeks after their third shot. And also that this does provide good protection against severe illness. Now, the big question is, why would this third encounter with a vaccine that is actually targeting the original spike protein work against this variant, which actually has more than 30 mutations within the spike? So I think some of the answers would lie with our cellular immune responses. So first off, with our B-cell immunity. Now we know with our experience with other vaccinations and viral infections that we would expect to have some degree of waning of immunity or waning of antibody levels. So take, for example, hepatitis B infection, the fact that we originally advised that persons would have to get vaccinated again or have boosters if their antibody levels fell below 10. So we know that this is a normal phenomenon and that we would expect this. But the big question is whether the decline that happens will actually stop and what the indications are. So we know also now that from more longitudinal studies that although there's quite a rapid decline from about 3 months to 5 months down the line after having COVID infection, that after that period, it seems to stabilize a bit. And the same has been noticed also with post-vaccination antibodies. So the levels tend to fall from about 95% to about 35% or 39% after 5 months. And although it seems like this means that the vaccine efficacy is failing, those persons do still have ability to protect against severe illness. So even though sterilizing immunity might not be, as effective anymore, there is still protection against severe illness. So the vaccines basically retain this ability, and even though the responses of the antibody levels are waning, our hospitalization prevention and severe illness prevention does tend to remain. So there is something that happens within the immune system, and it probably has a lot to do with B-cell immunity. So if we have a look, we know that after the initial infection, or in the case of vaccination, after your second dose of vaccination, you have your initial short-lived plasma cells, which actually produce the neutralizing antibody. But on top of that, these germinal centers also still are maintained. And even though the antibodies may die off, you will have these memory B cells that are produced, and long-lived plasma cells. And these actually will work closely with basically within your germinal center, and there will be some development and guessing within the germinal center to actually enhance the breadth of that antibody, or the way in which that antibody actually binds. So there will be maturation really of that antibody response. And when you do get a subsequent infection, you'll very quickly have mobilization of those B cells, and that will actually then help to produce more and more effective antibodies, which hopefully then will have a bit of a broader binding to the actual new variant. So the memory B cell response really has been shown to actually increase with time, and this probably also has implications then, as we saw with the duration of time between either our boosters or the duration of time from your natural infection to when you actually get vaccination. Then, of course, I'm not forgetting about the T-cell memory, but I'm going to focus on this a little bit later, because this probably also has a large role to play. Now, so to just get back to this, in a particular study, they compared then standard vaccination with either Moderna or Pfizer or Johnson & Johnson with those who were boosted with an mRNA vaccination, and then to show how effectively it would neutralize the virus variant spike protein. So in this standard dose group, it showed very limited ability to actually neutralize a Micron variant. However, in those who received a booster dose, they found that it was only about 4 to 6 times lower than what it was able to neutralize the original strain, which is quite a significant difference. So again, after 2 doses, we probably see the establishment of your memory B cells. And then after your 3rd shot, it actually prompts those memory cells to then become antibody-producing cells. And as mentioned before, these memory cell B cells really sort of go through a library of guesses about what the actual next variant might look like, and it actually is maturing that antibody and broadening the antibody response. Then, sorry, this slide I still actually wanted to take out, but this is actually the previous study, the New England Journal commentary that I was referring to earlier, was actually the update on that. So this was still before there was that data available, but this was basically also then where they showed between that polymutant spike that they produced and that the antibody levels were actually improved and had much better neutralization against that pseudo virus, or pseudo antigen, so to call. And had higher level of antibodies. So this is what we refer to as hybrid immunity. And hybrid immunity really just is basically in response to persons who had a natural infection and then had vaccination as well. And we see that these persons tend to have much better and more robust protection against variants. Having said that, of course, this is only what we know until now. And it might differ for future variants. But there are studies that show that, and I know that this is often a question, that if you've had natural COVID infection, are you not okay? And do you really need to then get vaccinated? We often get this question. But in fact, there are studies that show that the risk of reinfection is in fact 2 times higher in someone who had natural COVID but was not vaccinated subsequently, as opposed to those who did get vaccinations after COVID infection. So definitely a much broader immune response. So let's move on then to the cellular immune responses. Now, with the original SARS coronavirus, we know from studies that were done subsequent to that, that antibody responses were demonstrated to be relatively short-lived, but the cellular immune responses could be demonstrated for many years after recovery, up until about 10 years after the original SARS coronavirus. And it was also, in fact, shown to offer some protection against reinfection in animal models. Now, with SARS coronavirus 2, because of this experience that we had, a lot of studies also investigated the cellular immune responses, and they actually demonstrated that there is good activation of T-cell responses, and including memory T-cell response to various different proteins of the SARS coronavirus 2, and of course, including Spike. And it was also demonstrated that there are some reactive T cells in about 35 to 60% of persons who are COVID naive or seronegative persons who actually never previously had COVID. And this may well be ascribed to T cell recognition that might occur after exposure to other common cold coronaviruses, because these T cells tend to be more cross-reactive. So what we also know in general about the T-cell immunity, other than the fact that it also supports the B-cell responses and is necessary for B-cell responses, is that it's not as much associated with sterilizing immunity. So where neutralizing antibodies are crucial for sterilizing immunity, we do, however, know that T-cells are very important when it comes to protection against severe disease. And in addition to that, as mentioned, it's more cross-protective. And this is because we generally see that they have more epitopes, and these epitopes tend to be less prone to different viral mutations. So they tend to be less affected by variants. And it probably just is much harder as a result of that for the virus to actually get past the cellular immune responses. And of course, this is very important to consider when we're thinking about a vaccine design and have to incorporate a T-cell immune response and detection of T-cell immune responses. Then I'd like to show you that we've actually done a study of our own as well within AMPAT. We did a study to compare T-cell immune responses against the SARS coronavirus spike and the nucleocapsid protein, and we did this using an in-house flow cytometric assay. This was published this year in the Journal of Clinical Pathology. And it was a nationwide cross-sectional study, and we essentially compared the specific T-cell responses between lab employees who had and did not have previous COVID infection. And we detected both nucleocapsid and spike protein responses. So when we looked at a group according to their previous COVID PCR results, we found 81% T-cell proliferation and 62% as well in negative participants who also did actually have T-cell responses. And then when we looked at these groups classified then according to their COVID serology results, we found similar findings, a little bit higher percentage of T-cell proliferation found in this group, but also still 56% of negative participants were found to have T-cell responses. And also just to mention that the magnitude of the T-cell responses as determined by the stimulation index was found to be significantly higher in the group that had confirmed previous COVID infection as opposed to the negative groups. So the conclusion here was that a significant proportion of the participants who were not previously diagnosed with COVID did demonstrate SARS-CoV-2 specific T-cell reactivity, and we postulated, as was also shown in some other publications, that the T-cell responses may be attributable to cross-reactive immune responses, but it may also be the result of asymptomatic infection. And having said that, we know that the serology might not always be as reliable in the person who had asymptomatic infection because they tend to have lower antibody responses. So the T-cell responses may and have been found when they studied specifically the variants of concern to have much broader cross-protection as opposed to neutralizing antibody responses. And these two publications have shown this by Tark et al. They investigated persons following natural COVID infection with a pre-existing strain and also compared with then the variants alpha, beta, gamma, and epsilon, and also persons vaccinated. And they found negligible effects of the T-cell responses to all of these variants. And when they also, in particular, investigated the T-cell epitopes, they found that these were not really affected by any of these variant mutations. Also, Red et al. Showed similar findings, and they demonstrated that virtually all the T-cell responses were able to recognize variants of concern. So these were done prior to a Micron and was not including a Micron data. So when we look at a very recent publication, which was done by some of our local scientists, they did show that T-cell responses that were investigated in groups that had vaccination, either J&J or Pfizer, or unvaccinated convalescent participants, that the CD4 and CD8 T-cell recognition of the Micron spike was largely preserved when compared to the ancestral strain, and was also similar to other variants of concern. And they also found that the magnitudes were very similar within different antigens. So they concluded to say that most of these participants showed that there was target conserved epitopes in the spike, and this was despite the extensive neutralizing escape that we saw. So despite that, they saw 70 to 80% of the T-cell response was preserved. So this is all good news in terms of our cellular immune response, at least with variants. And other than our local publication, they have had similar findings also in Italy, where they also found that the Omicron was recognized by the cellular component of the immune system, and also that the presumption would be that protection would be retained from hospitalization and severe disease. And then this publication also basically the same findings. So just some further considerations when we take all of this into account is the consideration of doing mixed vaccination, which is also referred to as a prime boost strategy. So if we think about this maturation of our B-cell immunity that happens and the amount of epitopes that are targeted, the fact that we use different vaccines may well then lead to a broader and a more flexible and a more diverse immune memory response. And by adding more targets, it might actually also lead to better protection. So that, and if we know that, knowing that the current vaccines mostly just target the spike protein, we know that T cells can in fact then recognize the whole virus. So these, I think, should be taken into consideration. And although at the moment we don't have this for SAPRA approval for mixed vaccination strategies, we know that this is being allowed elsewhere, and the amount of evidence is still mounting in this regard. Okay, so let's just move on then to the actual laboratory testing and what consideration should be given to the influence of variants on the laboratory testing. Okay, so I always start off with this when I talk about lab testing and COVID, because I do think that this assists greatly when we decide which tests to use where and to understand the implications of testing. So the light blue graph would be your PCR test or the RNA response, which is obviously detectable at the time and even prior to the symptom onset. And then we subsequently have antibody responses that will become detectable, and also taking into account that here in dark blue is your antigen test, which in general is less sensitive and is more sensitive around the time of symptom onset until about day 5 after symptom onset. So if you have a Micron, and considering that the virus probably has a much shorter incubation period and a shorter duration before it actually moves along, it may also affect the antigen testing period. So these are all things to take into consideration. So just to summarize again, the test types, we obviously have PCR and antigen, which are our direct detection methods, but we also have antibody testing, and then I'll also talk about cellular testing, which was not included in this graph at the time. The PCR and the antigen, of course, being swabs, and the blood samples then for our antibody testing. Now, in general, when it comes to the variants and our diagnostic assays, we actually don't anticipate them to be affected that much. And having said that, we do investigate this, and the companies are also that send these tests to market to do extensive testing to look at the detection of these variants with their particular assays. But there are still investigations underway, and some of these are more difficult than others to actually perform. When it comes to the COVID PCR, one redundancy that's built into this is that most of these assays actually use more than one gene target. So in essence, the gene targets, because we're targeting more than one, the chances that all of these would be affected in one particular assay are very slim. And then this is also where the S gene dropout happened. Originally, this was seen with the Alpha variant, but now recently with the Micron variant as well, where we could see that the S gene was not actually detected while we were able to detect other genes. Having said that, the S gene dropout is not something that we would be able to detect in all of our PCR assays, because the PCR assays actually use different targets. So not all of them target the S gene, for example. So there's only certain assays where we would be able to see that S gene dropout. And that has anyway now fallen by the wayside, or is falling by the wayside, so to speak, with the emergence of the BA2 sublineage. Then in terms of the antigen testing, the good news here is that the majority of these tests actually target the nucleocapsid protein. And as mentioned, the nucleocapsid is not really the main target of mutations, and we don't see as many mutations, so it's a bit more conserved. And we have not really seen any major implications for the antigen testing. And I think it would have more to do with the actual kinetics of the infection itself, and trying to test as close as possible to symptom onset to optimize the sensitivity. We know that the sensitivity is lower than PCR tests in any case. And then in terms of the COVID antibody tests, so while spike antibody assays may theoretically be affected, we have not really demonstrated this thus far. And this has to do with how wide those epitopes in the binding is. It's more robust, and it's unlikely that there would be that much conformity to actually affect these. But this is also, of course, still under investigation and is monitored quite closely. That our routine laboratory diagnostic tests can unfortunately not distinguish between variants. So other than the nice fact of spike gene or fallout or dropout that we've seen with Omicron, other than that, that could be used as a proxy, but it cannot really distinguish between different variants in general, and it would have to be with a particular assay. So as a general rule, the PCR assays would not be able to distinguish. But as I've mentioned, it was seen as a sort of proxy that if we saw S gene dropout on a particular assay that we were using, that we could assume that that was a Micron at the time, based on what the genomic surveillance also showed. And again, as I mentioned, the BA2 lineage now, we don't see the gene dropout. Then the antigen tests, just to show again, there are quite a few publications out there, and also looking specifically at the ability of the tests to detect Omicron. And I know from local groups as well, where there have been investigations trying to look at existing antigen tests in use, that they have not really found any major ability or drop in the ability of these tests to actually detect the virus. And also just to mention that with our COVID antigen tests, because we know already that these tests are less sensitive than PCR, it would anyway be advised to confirm if there is a high pretest probability to confirm those results by a PCR if there's a strong suspicion that the person would in fact have COVID. So that was anyway a recommendation even prior to the emergence of Micron. Okay, so let's focus a bit on the antibody tests. And there's been quite a lot of development along these lines. As I've mentioned, we ideally would have wanted to test the neutralizing antibodies, but there are practical issues in that regard. So our commercially available antibody tests actually test for binding antibodies, and these are the anti-nucleocapsid or the anti-spike assays. And as mentioned before, these assays do demonstrate a good correlation, especially between the anti-spike antibody assays and neutralizing antibody levels. Having said that, we know that these do not measure the cellular immune response, and this always needs to be kept in at the back of our minds when we are interpreting these results. So, as I've mentioned before, the IgM and the IgG could appear from about 1 week after the onset of COVID infection. It could even occur a little bit earlier. But we can only reliably detect them from about day 14 onwards. So there might still be a development of antibodies from day 14. So these tests should not be used for the diagnosis of a current COVID infection. It can be used as a type of an additional test when there's a strong clinical suspicion and you have recurrent negative PCRs. But really, this should not be the main use for these assays. And also, just a word of caution that having said this about the IgM and the IgG appearance, it may, of course, then still be that the patient is actually infectious at the time of the appearance of these antibodies. So these tests cannot be used to make a call on whether the person has recovered. And there are studies also that show that there's variable findings in terms of the appearance of antibodies and the decrease in the viral load level at that stage. So it's definitely not a given that a person with antibodies would necessarily be non-infectious. And then just to note that the vaccine responses might take a little bit longer to develop. So these are best measured about 21 to 28 days after the last dose of a vaccination. So this is from our AMPAT chat that you are welcome to. I think the link would also be available under the offers, or alternatively, you can go to our website to download this. But basically, this just shows what we have on offer in terms of antibody tests at AMPAT. So we do have a nucleocapsid IgG, a spike IgG, as well as a spike IgM. And we have recently introduced a SARS-CoV-2 antibody profile as well, which basically combines those three assays. And the advantage is that it would then give you a combined interpretation of the results. So very similar to what you would see with for example, your hepatitis B serology, where you can actually then make or interpret the results according to whether it's previous infection or whether it's more likely a vaccine response. So the nucleocapsid IgG, just to mention that this is the sort of marker to show that the person had prior exposure to COVID. We wouldn't expect to have nucleocapsid antibodies in someone who had vaccination only. And this is because the vaccines basically just contain spike protein, or those that are in use within South Africa at this stage. The nucleocapsid IgG is not a quantitative test. It is a semi-quantitative test, or actually just a qualitative test. So that value that is reported on the actual report is not a quantitative value. It is basically just a value that helps us to know whether that response was above or below the cutoff value for the test. So to determine basically whether it's positive or negative. So it's not a standardized value that, and it cannot be used interchangeably with other assays. The spike IgG, however, that we do have on offer at this stage is quantitative assay. So I'll talk a little bit more about quantitative assay in the next slides. But this assay would basically be appropriate not only for those who had vaccination with COVID, but it would also be an appropriate assay to test the actual level of antibodies in someone who has recovered from COVID to get an idea. Then, of course, the spike IgM assay, which may enhance the sensitivity for the testing, and which is why we included it also in our profile. But previously, we only had this on offer as an individual test for travel purposes. Now, when we talk about the quantitative determination, quantitative tests have the advantage that they have very good sensitivity and specificity and very good precision. And in general, we find that these are very helpful to establish variation, not only across time in one individual, but also across populations. And in general, these assays have been developed to have a better dynamic range. So we don't actually need to do any further dilutions or anything like that. They are generally better for monitoring antibody levels over time. So as mentioned, either on an individual or for specific populations, for surveillance purposes. And the big advantage would be that you can actually have a better correlation of what the antibody response looks like, either between natural infection versus vaccination, or to just purely evaluate antibody responses following vaccination. Now, these assays have the ability to possibly be standardized with a WHO standard that has been developed. And this in future will help to reduce inter-lab and inter-assay variability. But at this point, the reference ranges are actually still unique to each manufacturer's assay. However, having said that, the aim of that standard eventually is to harmonize the assessment of these humoral immune responses. So very similar to what you find with your hepatitis B antibody level, for example, you would basically have a standardized international unit that it's reported in. And eventually this will probably be what will happen with the COVID antibody tests as well. At the moment, there's an option to convert it actually to binding antibody units, which is then based on studies that were done using the WHO standard. So, and having said that, this is not possible for the qualitative assay. So we can only do this for our quantitative assay, which would be the spike IgG assay that we have. Then if we look at the interpretation of these results, and this is again also from our AMPAT chat, so you're welcome to have a look at that. But yeah, just briefly, the reason we combine this is that it's quite nice to see if you're not sure in a patient and you don't know if the person possibly was exposed before or whatever the case might be, it's nice to have a combined interpretation like that. So if you have a negative response in both, obviously, most likely previous COVID is unlikely and no vaccination. But having said that, the negative result does not necessarily exclude past infection because a small percentage of people might actually not mount an antibody response. And as mentioned before, this will often be in persons who had mild or asymptomatic infection, and they might have had waning of the antibodies, of course, over time too. And then if there is a negative spike IgG, but a positive, a negative nucleocapsid IgG, but a positive spike, this is typically what we see after vaccine-induced immune response, whereas a positive nucleocapsid IgG and a positive spike would be seen in a person that had previous COVID infection, either then with or without additional vaccination. And then just a final warning, so to speak, is that we still unfortunately do not know what the actual correlates of protection are for COVID. So even though we are taking some strides in the right direction, and we're trying to have standardized assays and standardized reporting. We're just not there yet. So the information will be emerging as we go along, and they often get this sort of data from vaccine studies and vaccine trials. But there unfortunately hasn't always been a very standardized approach with testing following the vaccine trials, and some of these were purely based on the clinical outcomes of those trials. So as we go along, this information will probably still be established. But for the time being, we thought it best to actually mention this particular finding from a publication in our actual assay comments. And this also appears in our AMPAT chat. But basically, this is a correlation that was done between the spike IgG levels, and this is in the assay that we are using within our laboratory. And also correlating that then with neutralization values on a neutralization assay. So the positive cutoff for the antibody assay is 50. So anything above 50 will be deemed positive, but then you will also get an actual quantitative value in arbitrary units per milliliter. Below the value would be deemed negative. But if you compare that then to the neutralization values, so you will see the 50 actually correlates with only 18% of viruses neutralized. And that is actually a negative result on the neutralization assay. 30% is the cutoff for positive with the neutralization assay. So only at about 107 that they find that 30% of viruses were neutralized, or a positive neutralization. And as the antibody levels go up, of course, the percentage of viruses neutralized would also go up. So this will give an idea, an estimate at least, to be used in practice to get an idea. So even though we can't really say, okay, this is the value at which someone definitely has protection or does not have protection, you would at least have some estimate of where this person's antibody levels lie and whether it does correlate with neutralizing antibody responses or not. So the advantage is that it does offer the advantage of potential correlative protection in future as well, as opposed to semi-quantitative assays like the nucleocapsid IgG, which does not, unfortunately, offer that option. And I thought just to include this table from the same publication. This just shows you what the typical values are that you would see in persons who were vaccinated. So this was in response to an mRNA vaccination, I think specifically the Pfizer. Yeah. And you can see here by week 7, that is when the booster dose was given and what the jump in the antibody levels were. And just to note that there are 3 different assays that they used here, all of them anti-spike assays. But you can notice there and appreciate that there are various significant differences in the actual values between these different assays. So definitely it's not used interchangeably, but you could possibly convert these things to the binding antibody units per mol according to the WHO standard. Then also just to mention that theoretically, because of all of these mutations that I've shown, we would anticipate that it's possible that there could be some effect on anti-spike antibodies. But as mentioned, we have not really found this so far, and no studies have really shown any significant differences for previous variants, and so far also not really any significant changes for Delta, for Micron that have been noticed. So, but this is still ongoing investigation. Now, I thought before I lose everyone on this very theoretical topic, let's do some practical clinical cases to just demonstrate how to practically apply some of this serology. So the first case is a 40-year-old male. This individual actually had contact with a COVID case in December of 2021. He became symptomatic after a few days, but he did not do any formal COVID test at the time. And he recovered. He was, however, due for an elective hospital admission for elective surgery, and this was in January. When he did his pre-admission testing on the 11th of January, lo and behold, he had a positive PCR. So the question, of course, that arose was whether this individual was still having prolonged shedding even after recovery. And they basically wanted to then, or the hospital then advised the doctor to do an antibody test to be performed. So they actually performed the nucleocapsid antibody test, and you can see here the result. He was in fact positive then. Now, the question that they posed was whether the presence of the nucleocapsid IgG then implies that the person is no longer infectious. And so, of course, as I've mentioned before, the mere presence of an IgG does not necessarily correlate with absence of infectious virus any longer. So although we can look at the time period and the time frame from when the person possibly had the infection, we really could not actually use the nucleocapsid antibody result in itself to actually make this case. So one really needs to stick to what the rules for recovery are, and this would be based purely on the onset of symptoms. And hopefully, I know that after some time and discussion, they did actually accept this to be the case. So let's see, clinical case 2. This was a 57-year-old female. She had a Pfizer vaccination done. The first dose was on 19 May and the second on the 1st of July in 2021. Subsequently tested her spike IgG antibodies in November of last year, and she had a value of 992 on the spike IgG assay. Then on December, 29th of December, the person actually tested positive with a COVID antigen test. And she had symptoms at the time, which is why she tested, which was myalgia and a headache. And there were, in fact, also other close contacts who also developed symptoms and who also tested positive. She repeated the antibody testing after she recovered from the illness, and this was done on the 14th of January. And you can see on these results that here's your spike IgG, and there was a significant increase from the original 992. She's had a jump now to 39,000. And the IgM was also actually positive, so definitely a solid IgM response there. But the nucleocapsid antibodies were completely negative. And this is now the question, why would the antibodies be negative if the person actually had COVID infection and significant enough to be detected by an antigen test? So one could make a case, I suppose, to say that it might be that the antibody response was just not detectable by then, but we would expect to have a detectable antibody response by this time. It was at least 14 days later. I think probably more likely that the immune response was following the previous vaccination, and that she actually had a very good, robust B-cell immune response, memory response that then kicked in and then had a very massive amount, of course, of the spike IgG that was developed. And as a result of that and this neutralizing antibody response that kicked in very quickly, it probably just led to a substantial effect on the virus and actually neutralized, and that the body just did not need to actually have an antibody response mounted against the nucleocapsid nucleocapsid proteins. So that's I think that this is most likely the reason for this, but just goes to demonstrate that we don't always know what the kinetics of the antibody response is following vaccination. And then clinical case 3, this was a 54-year-old female. The lady had COVID-19 already in June of 2020. Then at the end of last year, on the 20th of November, she did a COVID antibody test. And she did the spike to get a quantitative value. And you can see here it was 2,708. And so the question was whether this person can use this result to motivate to either get a vaccination or not get a vaccination based on this particular result. So I advise that we do not know what the correlates of protection are. We certainly do not know what the correlates are for variants of concern, and in particular Micron, which we know is highly transmissible. And in addition to that, a possibility of immune escape, we know from the literature and the discussion around hybrid immunity that you probably would get a much broader immune response. And as mentioned, individuals who had natural infection had a 2 times higher risk of a reinfection as opposed to when they had vaccination after they had this their infection. So my advice to this person was still to get a vaccination regardless of the antibody result in this particular case. Then a published case report that I just found interesting. This was a 43-year-old male. He had symptoms in March of 2020 and tested COVID positive on the next day, was hospitalized at the time. And then at the 9-month follow-up after his recovery, he actually did not have any detectable neutralizing antibody responses any longer. Then after, or between this time when he was actually monitored, he suddenly had a sharp increase in his spike and the nucleocapsid antibody responses subsequently. So you can see here there was a significant increase in the levels. And this demonstrated quite a strong anamnestic response and probably a strong recall response with these increased and high values. They also then investigated this person's antibodies and had a look at whether or not it was able to recognize different variant spike RBDs, and they could demonstrate that it was very good recognition actually. So this person must have had a reinfection at this point. He did not have any boosters or vaccinations or anything like that. But he actually remained asymptomatic. So despite that decline in the neutralizing antibody response, he most likely had a very good memory response that then kicked in. And finally, case number 4, we had a 23-year-old female. She was unvaccinated and did not have any prior documented COVID. And then in December, the 3rd of December, she tested COVID positive, had very mild symptoms at the time, only a headache and a bit of a sore throat. The person then tested their antibodies on day 8 after the COVID infection, as well as on day 35 after COVID. And these are the results. So you can see here, there was no IgM response at all, even on the later bloods from day 35. And also no spike IgG that was detectable. She did, however, have nucleocapsid antibodies positive. However, these were quite low values. So you can see it was just over the cutoff of the assay, 1.67, whereas the cutoff is 1.4. And then following this, she decided then to rather have a vaccination. So she had her COVID Johnson & Johnson vaccination on the 17th. And then she repeated her antibody profile again on the 31st of January. And here you can see what the subsequent results are. So there was still no detectable IgM response for the spike protein, but there was at least a good IgG response where it jumped to 1,200, and the nucleocapsid remained the same, which is not surprising. So again, just to note that the antibody kinetics are not always clear-cut. The spike and also the nucleocapsid versus the spike kinetics may also differ. We also know by now that there's various publications also that demonstrated that the nucleocapsid IgG might actually appear a little bit earlier than the spike IgG. And then it might also wane a bit faster than what the spike IgG does. So there definitely are differences in the antibody kinetics between the different targets. And we also know that a mild infection may be associated with much lower antibody responses, which may very well be the case here because she had very mild symptoms to start off with. And I think also just important here to show that if a decision was taken purely based on the fact, and if she had only tested her nucleocapsid antibodies, and she saw that, okay, she had an antibody response and just let it be, she probably would not have had her vaccination. And that probably would have left her vulnerable to a possible reinfection again. So again, showing the value of doing various antibodies and not just one of the antibodies, as we offer with the profile. So just to summarize the antibody responses briefly, we know that reinfection antibody kinetics is still not entirely clear-cut. We don't exactly know what post-vaccination breakthrough infection antibody kinetics look like. And we also don't know exactly what the antibody kinetics are following different infections with variants. And it's becoming very complicated to actually investigate this because we are having more and more mixed infections, people who have had previous COVID infection that actually have reinfection, or persons who had vaccination and had breakthrough infections. So it is becoming a bit of a complex field to get clear-cut answers and needs further investigation. Okay, then to move on to our T-cell responses. So for laboratory testing, we do offer T-cell response testing. And we offer two different T-cell tests for COVID. We offer one that includes both the nucleocapsid and the spike proteins, and we also have a spike only T-cell test. These tests, just take note, because these tests are done and it's done over time, so it takes about a week to complete, and it takes some time for the processing to be done, and the laboratory it does not function over a weekend, so it's important to collect the samples only from Sundays to Thursdays. What we do is these tests are then expressed as a stimulation index, and this basically gives you a ratio which then compares the original patient's T-cell response with COVID proteins and then without COVID proteins. And also, as mentioned earlier, there may very well be cross-reactive T-cells that can also be detected during this, that might be the result of prior infection with common cold coronaviruses. So ideally, these tests should be interpreted in conjunction with antibody test results to get an idea of what the patient's cellular immune response looks like. Then I thought just to very briefly, and I'm not going to go into much detail on this, but while we are talking about variants and antibodies, I don't want to leave out the issue of monoclonal antibodies. So although this is not something that is really available yet, it is something that is being applied overseas, and the monoclonal antibodies generally are used for mild to moderate COVID infection in persons who are at high risk of being hospitalized. And the WHO have actually approved some tests. Conditionally, they have approved sotrovimab, which is the last column, and this in addition then to a previously approved drug or recommended drug, which was a combination of casirivimab and imdevimab. However, it should be noted that some of them are affected. So you can see here in this table, some of them are affected by the variants and some of them are not. So it's also important to take this into consideration should we in future start using these in our setting as well. And then just finally, I did not make much mention of the innate immune responses, but this particular study was quite interesting to me, where they demonstrated that Micron is actually more sensitive to inhibition by the interferon response, and that this in fact may be a possible explanation as to why the variant is considered to be less severe clinically. At the same time, they also investigated the Micron response to different antiviral drugs and drug candidates that are in the pipeline, and they found that it remained that these drugs remained, or that Micron was still sensitive to these particular drugs at that time. So just something interesting to note. So after all this, just in conclusion, I think it's important that we note that we would need to continue to do genomic surveillance. And we need to try and do this to monitor for any possible changes and to do that as soon as possible prior to the emergence of anything that might have severe implications. And this, of course, still needs to be shared internationally. There needs to be continued efforts also then to investigate what the effect is of those variants on the ability of diagnostic assays to either detect the virus itself or to detect antibody responses to the virus. And furthermore, just to note also that the quantitative antibody assay, spike assay, we know correlates very well with neutralizing antibody responses. And despite that, the fact that we don't know the exact correlative protection, this assay may be very useful going forward to correlate immune response between natural infection and vaccination. And it's also a step closer to standardization because of the WHO standard. It should be kept in mind, though, that the antibody tests will only provide clues as to the humoral immune response to COVID, and that cellular immunity, which we know may be more durable because of the T-cell epitopes not being affected by the variant mutations, that this should also be investigated and kept in mind. And of course, we still need ongoing investigation into what are the antibody kinetics that we expect to see after reinfection, what are the post-vaccination breakthrough infection antibody kinetics, as well as antibody kinetics following infection with variants. The lab testing of all these immune responses, and by increasing our lab testing, we will contribute then to our understanding of these responses more and more. It's becoming increasingly complicated really to know because of the proportion of people who have had prior COVID infection and vaccination in the population. And I think that vaccine design really needs to aim for broader and more cross-protective immune responses. And there is a lot of talk about variant-specific boosters that are in the pipeline. And taking into consideration that there's some preliminary studies from Israel that showed that the fourth booster shot does not necessarily have as much of an effect to protect against variants as what the third booster dose has. There is a lot of talk now about going for variant-specific boosters going forward. Yes, and that is the end of my talk. Hand over to Jan-Rie again to discuss and to have some questions.
Thank you very much, Dr. Marieke. It was very insightful, and I think everyone really gained a lot of knowledge by listening to your talk. So I think we have a few minutes for 1 or 2 questions. There was a few questions. If we don't answer your question tonight, I will send over the questions to Dr. Marieke, and she will try to come back to everyone. But the one question I think we can have a look at, do you have any comments on the notion of T-cell fatigue arising from excessive boosting, a possible argument against too frequent or too repetitive boosting? Under the question and answer box.
Yes. So yeah, I do think that we really need to be cognizant of the fact that there is an optimal time frame for the development. And we can see from these, I think not only because of the concern about T-cell fatigue, but also because you just want to have that optimal response. So to allow the B-cell memory response to sort of, go through its natural process of optimizing the actual antibodies that are developed, there probably would be an optimal time between boosters. And I think it's difficult because we often get questions from people because they want to travel and they want to know if they should rather have the booster earlier and should they, try to do this before the next possible wave. And people are concerned that they've got very fragile family members who they would want to boost earlier rather than later, or get the timing right between waves, or whatever the case might be. But I think, as I said, we are still learning, and there are, changes as we go along. And I do think that, there are there's a lot of publications to show that probably having optimal dose or optimal intervals between those doses would probably be more advisable. And there are some studies also that looked at just the antibody responses and how much it gets boosted after further doses. And there wasn't really much of a difference between the antibody levels that were that developed following further boosters down the line. So we don't really know if there's much of an advantage at this point. So I would advise against just having a monthly booster, so to speak.
Perfect. Thank you, Dr. Marieke. And then another question we have is there any reduced risk of transmission as a reason to reduce isolation and quarantine times? I assume the person asking the question should refer to Omicron. Yes.
Yeah. Yes. So we know that the incubation period has been shown to be shorter, and probably the duration of infectivity is also a bit reduced. But I do think that we also need to consider that a lot of the recommendations at this stage are also, a move towards the next phase. And I know that the WHO doesn't want to use the endemic phase, but to transition from this being sort of the acute infection or acute pandemic phase to endemic will also require some changes. And I think just the futility of how infectious this Micron variant is, and trying to actually, do contact tracing when from a practical point of view, it's just not possible to do. And also, I think there's been a lot of literature to show that there's been a lot more mild and asymptomatic infections as well when they did, in surveillance testing. So it just becomes almost impossible to really follow everyone up. So I think that, it probably stems mostly from there that a lot of these new recommendations have come about, and not necessarily only to do with Omicron itself.
Perfect. Thank you very much. I think that's all the questions for tonight. So I'm going to share some files on the right-hand side so everyone can have a look at that. And there's also an offer which you can sign up to receive regular and important information from AMPAT, including the data around our respiratory viruses and the detection rates of those. So just to conclude the event tonight, thank you once again, Dr. Marieke. It was a great presentation, and a reminder for everyone to explore the file handout section to download our COVID-19 testing options pamphlet, and also have a look at the antibody AMPAT chat that Dr. Marieke referred to. And I also just want to mention, if you completed the survey for CPD points upon registration, and I see a whole lot of attendees also posted their HPCSA number in the chat box, thank you for those. I will make sure that you will receive 2 clinical points for attending this session tonight. And thank you again for attending, and have a lovely evening further, and goodbye.